All right. Hello, everyone. Thanks for coming to this month's Ag Bio Data webinar. We are very pleased to have our speaker this month, Moni Munoz-Torres, who will be telling us about the Monarch Initiative. So Moni and I actually go back quite a ways. I was reviewing her CV. So we started out as postdocs together at Georgetown University in Chris Elsick's lab when Moni was working on the Hymenoptera genome database. And she's done a bunch of other things since then that I've been able to catch up on. So, but I should say currently she is an associate professor at the Anschutz, University of Colorado Anschutz Medical Campus. She is the program director at the Translational Integrative Sciences Lab. But before she got there, she was actually very well known as the program manager and curator and bioinformatics scientist for the Apollo project at the Lawrence Berkeley National Lab, where many people confused the two of us when I was working at the I5K workspace, and we would often get emails to the other person and had to say, no, you mean the other Monica. So don't confuse us now. After that, she had a stint at Phoenix Bioinformatics. So she's known by several people on this in this group as well. And after that is when she moved over as the director of the Translational Integrative Sciences Lab. So she has a lot going on. She's been a member of many different committees. She's a very interactive scientist, is on, I believe, at least she was very active in the International Society for Biocuration. You might still be, apologies that I don't know that, but she was the chair at some point and very instrumental in that committee, also in the I5K coordinating committee back in the day for insects. And so today she's going to tell us about the Monarch project, which is she's currently the program lead for, I believe. And this was actually prompted by a webinar a couple months ago by Chris Mungle, who told us about the Gene Ontology Consortium as a way to sort of integrate gene function across species comparatively. And it came up that, yes, maybe we need to have Moni talk about the phenotypic and phenomic side of things in the Monarch project. So I'm hopeful that we're going to get a lot of great discussion, since this is an issue that really affects our community as well. So Moni, go ahead and take it away.
Thank you, Monica. It's just, I'm absolutely thrilled to be back with you. I had the privilege of being part of Ag Bio Data in its inception, and I'm delighted to see how far you've come as a team. And thank you so much for that introduction. As Monica said, I work at CU Anschutz, and in there, I'm the program director for the Monarch Initiative and the Phenomics First resource. The director of the Translational and Integrative Sciences Lab is our fearless leader, Melissa Handel, and we have a team of about 26 people with 3 tenure-track faculty who work together in a matrix-like system. We are a little crazy and love it that way. So I'm going to start sharing slides now, and I'm going to tell you a story about the Monarch Initiative. There we go. I think you should be able to see my slides. I'm going to hide the controls if you give me 1 second. And goodbye controls. Okay, thank you so much. So the first thing I'm going to do is introduce you to the Monarch Initiative, and then I'm going to walk you through information about our tools and resources, and then I'm going to tell you a little bit of detail about some of them, and hopefully at the end share, leave you with a taste of where you can go find some of these resources. We're both a consortium and a resource, and we basically look into bridging the space that exists between basic and applied research. A dear collaborator of ours calls it the chasm of semantic despair. And what we're trying to do is developing tools that facilitate this connection across all of these fields using semantics-based analysis. So we're focused on the interoperable aggregation of data from humans and many other organisms, and on the development and implementation of ontologies and tools and standards so that we can build that bridge into clinical cases. And so I'm going to showcase a few of the use of model organism data for diagnostics and for establishing genotype to phenotype connections. And I hope that you will accompany me in this journey and learn a few things about what we do, and then we can have a conversation about how we can help. So it starts with, the way in which the data are organized. For us, was of concern, of course, when data are distributed, they can be very rich and have high quality, but unfortunately, the conceptual frameworks can vary for the aggregated data, and so it's a little challenging to be able to parse it. When we have species-specific or data type-specific lines of inquiry, you can answer those questions with distributed data. The next step, I guess, is to aggregate the data that you had distributed like this. And so we have the ability to enable large-scale indexing, and then this could be used for general search and retrieval. But I guess the best way to put it is, to put the actual picture together, the metaphorical one and the actual one in this example, we are data integrators. So we align the distributed data so that we can share conceptual frameworks, and we use consistent identifiers in order to do so. And it allows us to look at the big picture, to detect where the gaps exist, and to do derive insight across domains using both curators and algorithms. And so both sides of this chasm of semantic despair are not so very different, right? And ultimately, in both ends, if we're looking at clinical things and we're looking at the basic science things, we're both looking at, the observation occurs from the clinician or the researcher on the patient or the specimen through documentation in both cases, then towards coding things in a clinical coder on the clinical side, or by curators' work to produce the information that goes into some sort of coding system, a code system in the clinical side, and an ontology on the research side, and eventually go into some type of data storage. And then the question becomes, how do we integrate it? And I'm here to tell you that given our experience and the things that we do, we are convinced that the answer is actually semantics as the ultimate universal converter. And so bring on the ontologies. I know that many of you work with ontologies, so I will not take the time to explain what those are today, but rather just share how, from the perspective of the electronic health record on the clinical side, there's all of this information that is coded into standard terminologies. The problem with those is that they're usually less computable, and they have slow development cycles. It takes forever. The community is not really involved in developing these private efforts. And there's a lot of, the release cycle is long, so there's rigor in that process, lots of tools, lots of mappings. While on the research community side, we have the ability to work with rigorously computable ontologies for modern AI applications and cycles that are much faster. However, they are constantly changing and can be of lower QC processes. So the idea is to make sure that we meet in the middle. And so I'm going to tell you a little bit about some of the answers that we have found in order to continue to bridge that gap and find in the middle. So we'll start with clinical phenotypic and diagnostics, and sharing that this actually requires the ability to leverage multimodal data in modern classification strategies. And so when as I was saying earlier, when knowledge is distributed, the diseases concepts don't really align. In this case, if we're investigating, a while back, we were looking at the representation of something called the Zellweger spectrum or disorder. And different disease resources had different ways of labeling the information. They had different labels. The terms had different parents, different children, different synonyms, and different textual definitions. And so the question was, are these actually entirely equivalent? So if different communities annotate different relationships at different levels of granularity and using different vocabularies, several different disease terminologies end up existing. And so, and as well, terminologies that help you describe other aspects that are relevant to human disease, like, as you see up here, gene variants or phenotypes and exposures. And so a single terminology that can bring all of these concepts together and bridge that gap between all of these disparate types of information would be very useful, which is exactly what we got busy working on. So we decided to work on a Bayesian approach that would bring merging equivalent classes or equivalent disease concepts using the evidence and utilizing logical and probabilistic inference in order to be able to create a disease ontology that integrates all of these concepts and resources that you see here on the left, among others. And our team of curators actually ensures that the resulting equivalence inferences are accurate and are supported. So we also make this an iterative review process and provide feedback to the various sources and the disease data. All of this results in what we call the Mondo disease ontology, which is a logic-based structure that harmonizes disease concepts and phenotypes across species, actually. And on the right, you can see an example of the representation of the concept of endometriosis. And this is a disease characterized by the growth of functional endometrial tissue in anatomic sites outside the uterine body. And you can see something that is probably very familiar to you, a tree view of the classification of the disease as, you know, a disease of the genitourinary system with reproductive implications, and all of this information here in some more details about the children terms in here. Importantly, we also provide mappings to this term to all of the equivalent sources that were merged. And so the medical subject headings for thesaurus, the NCI thesaurus, codes from the International Classification of Disease terms, and also other terms are also available. And so the representation of disease with an ontology ends up looking something like this. This is an example of how the resulting representation of a disorder looks like in the ontology. This is REFSOM, adult REFSOM disease, which is a very rare, clinically variable, multi-systemic metabolic disease. And it can be represented as a Mendelian disease, a metabolic disease, a nervous system disorder, and there are all of these subclasses about the disorders of the central nervous systems, genetic nervous system disorders, et cetera, et cetera. And so this is all of the representations of the adult REFSOM disease. So in addition to developing the Mondo disease ontology, we also developed the Human Phenotype Ontology. And so this is machine readable, also standardized, and it's a vocabulary that encompasses all phenotypic abnormalities that we encounter in human disease. There's over 14,000 terms. And we use these to create computational disease models. And so each term in HPO is interlinked with other ontologies, many of those familiar to you, such as, this hyposmia is connected to the sensory perception of smell term on the gene ontology. There's also terms to the cell ontology, as you see here on the motor neurons, and then the Monarch disease ontology is as well, which is the one that I just mentioned. And so the information in the Human Phenotype Ontology has computational definitions for each of the classes in the ontology. And whenever possible, the diseases are actually annotated to phenotype terms from the HPO. And the more specific the term, the fewer diseases are going to be annotated to it. It is important for us to think of also identifying phenotypes in species outside of our own. So other species aren't just relevant, they actually have unique proteomes and phenotypes that we can utilize and leverage. And so, great pond snails are models of inflammation-mediated memory dysfunction, and they show evidence of spontaneous neural tissue regeneration after injury, which is, of course, of interest for us. And silkworms are a model for uric acid metabolism, and it is known that decreases in the plasma uric acid are correlated with clinical progression of Parkinson's disease. And cats and dogs and shrews and armadillos are also very relevant models for human disease. So in this picture, what I've done is counting the phenotypes for human genes curated in humans over here in the, I think you can see my cursor, in the green circle on the left with the 4,000 number. And they represent only about 21% of the total number of protein coding genes that we know in the human genome, given the HGNC version in October. And on the right, we see the availability of all the curated phenotypes of abnormal traits in other, this have been attributed to human genes. So these have been either directly inferred, the annotations have been not directly, or they have been inferred through ethology. And this is across all of these very familiar model organisms. And we can see that data from just 5 species raises the phenotypic coverage of the human coding genes to 84%, from 21 to 84%. So truly, the model organisms provide us with key insights into the phenotypic manifestation of coding genes. And so we have gone in this direction, exploring the anatomy of other species as well, in order to be able to get to a place where we have more information that allow us to leverage everything that we know about model organisms in order to better represent and understand the knowledge that we have for ourselves. So Uberon is another ontology that we develop, and this is an anatomical ontology. It represents body parts and organs and tissues in a variety of animal species with a focus on vertebrates. And it integrates multiple anatomy ontologies into a unified one that is interoperable across different species. And it can really easily generate different views of different taxa and domains, like the respiratory system that is demonstrated here, or context like, data collection forms, etc. So this is one of the ways in which we leverage that knowledge by organizing anatomy concepts. And we also organize the annotation of phenotypes across species in a manner that is analogous to the unification that you're familiar with, that happens on the gene function annotation that the gene ontology does, right? And so uPheno builds on efforts that directly leverage the work of the phenotype ontology community and incorporates phenotypes from a much wider range of species than before. And this allows us to do the phenotypes from mouse and human here described, mouse on the left and human on the right, are unified under the uPheno terms, which are highlighted here, right? So the renal hypoplasia in human is unified with the duplex kidney phenotype in mouse through the abnormal kidney morphology. And we go down the phenotype collection in order to be able to create these profiles that allow us to do fuzzy matching across the species. And this helps us, at least in our case, to improve diagnostics. So wonderful things happen when we combine our work on data and ontologies and tools and algorithms and standards so that we can facilitate diagnosis, right? Ideally, our understanding of all of these pieces of data are going to help us narrow down the number of candidate diseases when we're trying to come through the information that we get from exomes and whole genome sequencing efforts. And the majority of the clinical diagnostics pipelines, much of the available data is basically underutilized. And this diminishes the power that we have to discern about these candidates. If we combine the genomic data and the phenomic data in the computational evaluation of these exomics sequences, and we use, in addition to that, human and model organism phenotypes, then we improve our ability to prioritize and enable more diagnostics. So this is done with one of our tools called Exomizer. And I know that I'm flying through some of these concepts, but I would be delighted to give you a little more information about what Exomizer does and how it has supported some research. And so as it ranks candidate variants based on patient phenotypes, we know that this supports the idea that the developmental history and the anatomical homology relationships all matter when you want to compare gene function or dysfunction across different species. So this is an example of how this was used. This is an interesting new gene candidate for congenital cataracts. And so we see that there was a rare predicted pathogenic missense variant in the mother and daughter pair in the CDKN2A gene. So the Genomics England has a series of panels that they run, and this particular variant was not in their cataracts panel. And there was no evidence in the literature, in any of the published literature and in the databases for this to be related to any human diseases. But using Exomizer, we actually, based on a phenotype match to a mouse that was produced by the IMPC Consortium, we were able to show a top hit, and this mouse also had abnormalities in the eye, including cataracts. And so in conclusion, it is, in order for us to be able to conduct this good diagnostics, we have been able to leverage all the knowledge that we have from the model organisms. Where one of the glitches is we need to be able to represent and utilize all of these phenotypes in computable ways. So this takes us to the next one on the set of tools that I want to share with you today. So for us to be able to properly conduct these discovery efforts, it was necessary to have a standardized way to represent phenotypic abnormalities, right? So you are very, very familiar. You know that there has been standard exchange formats for genome sequences that have been in existence for a long time. But for phenotypes, they didn't exist, at least not in a way that allows us to constrain the data to a model that is research ready. And so we desperately needed one such type of mechanism to share case level phenotypic information, improving the use of only the PDFs or downloading the entire electronic health record in order to be able to use it. So we created one such standard and we called it the Phenopacket. Did not kill ourselves with a name. It's basically literally that, a packet of phenotype data that can be used anywhere and can be written by anyone. And it's an exchange format of phenotypes and environmental factors as well. And so they encode ontologies and can be used to relate a patient's phenotypic profile to model organisms. And they can help us improve diagnostics. And they allow us to exchange information about the patient, the identifiers or the age, as well as you see here, the details about each of the observed phenotypes, whether or not a phenotype was observed, when they were first observed, how they were observed, and how severe a phenotype is, and how each phenotype is linked to a patient or to their genomic information, to the samples that were collected, and when available, to the patient's parents and siblings. And we actually, with the times ended up in the latest version of the standard, which was released in July of last year, we included improvements for the representation of cancer and COVID-19 phenotypes as well. Like you, this was absolutely and completely a community effort. All of the requirements and specifications were established with a community of researchers and clinicians, and it underwent a very rigorous peer review and product approval process. The first version was released in 2019, and then, like as I said, the last one in 2022, not 21, in 2022. But all of these with feedback that we received from the community, so we could expand on the data model and better represent, temporality in medical actions and other things. So this is the schema has a number of optional elements, and so you can see here, you can define the individual, the phenotypic features, its measurements, biosamples, any interpretation of the information that you receive, diseases, associated medical actions. But this is not exclusive of representing phenotypic data in humans. You can also, of course, represent phenotypic data about biosamples collected in veterinary animals or just non-human animals. So you can represent this information as well. And so the short URL over here is the slides again, so you can go to them, and then you can get this longer URL where we have more details about what a phenopacket is. In general, we think that the representation of data, of phenotypic data into an exchange format like this is going to benefit everybody, research biologists, physicians, patients, because they can now allow us to support diverse users and use cases in the context of the biomedical ecosystem. We know that there are many different providers of phenotypic data. I don't know, thing is working here, but including the patients and the clinicians, and they're collecting data through a variety of different mechanisms, including mobile health gadgets and, as you see here, the electronic health record. And the Phenopacket schema acts as a common model that can capture the data from many sources with a unified software representation, and then it can be used in by multiple receivers of the phenotypic information. And this includes journals and databases and registries. And their implementation can improve the speed and accuracy of diagnosis, as well as the treatment and effectiveness. This is work done in the context of the Global Alliance for Genomics and Health. And it is actually the very first ISO approved standard from the Global Alliance. We're very proud of that. It was a lengthy process. So hit me up if you want to learn more. You can also visit phenopackets.org. So all of this takes us to this concept of being able to map concepts across ontologies, right? Like keep saying that we're leveraging across. So mapping requires that there are curation rules and provenance and evidence and versioning and attribution. And I'm preaching to the choir because I know this is exactly what you work for. And so we, understanding that there are several different ontologies, we decided to come up with a standard that would allow us, a schema that would allow us to connect the dots across ontologies. So there's a range of mapping types across different ontologies that are possible. So we can build mappings across relationships like different entities related biologically that are curated relationships provided by authoritative resources, or you can have relationships that connect entities that are different but represent an analogous concept, or identical concepts, or relationships that are brought to narrative growth. In this case is an example of how Fanconi anemia can be represented by integrating information about ontologies that describe diseases, phenotypes, genes, and proteins. So this mapping model, we call it the simple standard for sharing ontological mappings, pronounced SSSOM. And this was created to harmonize the ontologies that are used for a variety of resources. And so I would love to go in more detail, but I know that we have a limited amount of time. So let me just say that, being able to mapping the evidence and the provenance and the attribution are all things that can be maintained through the process of implementing SSSOM as a model for mapping and communicating all of the information of these ontologies. So now you start seeing a little bit of what is happening and how we're constructing this history of building an aggregation that is interoperable of knowledge from a variety of resources. At the heart of, my apologies. I missed this bit. I forgot that I left this in here. I'm going to skip over this just to share that we get to this moment of at the heart of all the resources of the Monarch Initiative, we have this sophisticated ontology-based knowledge graph of genotype to phenotypes associations across a variety of species, from humans to classical model organisms to many less of those well-served species. And so the knowledge graph for Monarch combines information from dozens of species from, more than a dozen species from dozens of sources together with their ontology annotation. So you can see, about 800,000 gene to phenotype associations, and from about 126 species, and also 9,000 associations that are causal from gene to disease and non-causal associations for gene to disease as well. So genotype to phenotype associations and other data across a variety of species. And you can visit monarchinitiative.org to learn more about what is available. But we can leverage the information that we have on the Monarch Knowledge Graph in order to conduct a little bit of machine learning with several different use cases for variant prioritization, like I was showing you earlier, represented here on the left. And if you have an orphan disease, we want to know what causes it, right? Like, so we take any disease with no known causal gene identified, and we do the process of being able to embed the information that we have on this graph in order to be able to better identify the closest genes to closest genes to this disease. So the Monarch Knowledge Graph allows you to hold rich data from a variety of species, and it can affect it. And if you are doing graph machine learning on this graph, you can capture everything that is available, as well as characterize proteins that are not so well studied by leveraging the information that we have from other species. And so we can, in this very busy slide, I'm basically sharing that, say that you take any diseases with no known causal genes, and then you can use graph embedding in order to allow you to identify the closest genes to this disease. And they're close for a reason. You end up finding them in close vicinity of each other. And then you give the opportunity to the researcher to now dive with biological principles into the causes for that relationship. You can take every disease where there is a drug that treats it, for instance, and this is a direction in the latent space that exists from one to the other entity. If you do this for every single disease and every gene, you can eventually find the related gene to that orphan disease that you started with. You can, these knowledge graphs have tons of nodes, and they have millions of can have tens of millions of nodes. And so what you do through this embedding process is basically reducing the size of the knowledge graph, in this case of the Monarch knowledge graph, so that we have a lower dimensional space, so you don't have to worry about what kinds of, how long is it going to take, or how much memory you're going to have to use in order to be able to run your experiments. And so what we're demonstrating here is that in the, say we have 2 dimensions, if we are doing random walks to sample the neighborhood to reduce the dimensions that we had in our tens of millions of nodes into just 2 dimensions, as we're showing in the embedding over here in this little color. And that closeness and that latent space that exists across those concepts, it's going to have meaning because it's using the graph to put them closer together. So say a pink dot representing a gene and a green dot representing a disease, which I know this because I've looked at this image for a very long time. There are some of them actually here. So that is a meaningful expression, and you want to give this to your researcher, producing this information so that they can go and dive deeper into what are the true links of that particular association. So using semantics in order to integrate all of the multiomics data that exists is essential for understanding the molecular nature of disease processes at all stages of the development. In these genome readouts, so phenomics and metabolics and proteomics, transcriptomics, genomics, can reveal diagnoses and immediate therapeutic interventions. So to conclude, in almost every disease area, model organisms have been used for decades to reveal the causal disease mechanisms, right? So, however, as much data as has been produced, the vast majority of the organismal data is still underutilized, both in clinical applications and in biomedical research. And this is due to the lack of fairness of the data. The data is shared, when actually, if it is shared at all, using heterogeneous formats, heterogeneous identifiers, schemas, etc. And it continues to be really challenging to interpret and integrate these data at different biological levels, from the phenotype to the environment. And so the Monarch Initiative is basically a first of its kind semantic data integration platform with a focus on cross-species genotype to phenotype and disease data associations that supports hypotheses and the exploration of mechanisms that can be done by both clinicians and the basic research community. And Monarch also provides us with critical services and artifacts that allow for the diagnostics tools to be employed and other third-party platforms to be using it. Some of them you might be familiar with, like ClinGen and the NCATS Biomedical Translator, NLM's MetGen, the Genetic and Rare Disease Information Center, GARD, and the European Bioinformatics Institute and CPI, Kids First, Genomics England, and ultimately increasing the fairness and the trustworthiness of the Monarch resource is going to help us ensure that the data is a lot easier to access, a lot easier to utilize, to employ and implement into downstream analytics, which can include artificial intelligence and machine learning as well. I am delighted to have been able to share a little bit about what the Monarch Initiative does. I hope some of you see yourselves represented here, as these are current sources of the information that we leverage in the Monarch Initiative. And on the left, a list of all of us in the Monarch Initiative Consortium. And thank you so much for being here this afternoon and listening to me share a little bit of what we do. And I would be delighted to take some questions.
Thank you so much, Moni. This is a really interesting talk and a lot of information. I'm still sitting there to process some of it and have some questions, but excellent. Looks like there are some questions coming up, so I'll read the first one from Meg, but anybody else, please feel free to either post your question in the chat or just raise your hand and we can take your questions that way. So Meg is asking how much computer resources are required for a project like Monarch, and how much could be reduced by efficiency tweaking?
Cheers. Thank you so much. A lot. So we no longer have any physical computing resources. It's all now Google Cloud Platform. And I will have to fail the test of telling you the exact number. But the reality is that because we have been able to, in order to produce all of the knowledge graph, we have been going through a very extensive process of modernization of how the knowledge graph is constructed, as well as refining the biological understanding of each one of the sources that we're taking in. And so that in itself is reducing the size of such ginormous graph. If you're trying to look into a knowledge graph that has billions of edges, it can take you about hours in order to be able to get through the entire knowledge graph. And so we have been using these technologies to reduce the number to maybe 3 hours in order to get through the entire knowledge graph instead of days in order to be able to run it. And in that process of embedding considerably reduces the amount of compute time, in a logarithmic way in order to be able to make better inferences utilizing the resources. I will have to get back to you on a specific number of how much we have on our GCP.
All right, thanks, Moni. Tanya, how about you?
Hi, Moni. I was wondering about Phenopackets for plants. So right now, great application for humans, possible extension into organisms that use Uberon and CL. Maybe you've already thought about possibilities for plants, and if so, could you share? And if not, what would you advise?
Absolutely. So Phenopackets are species agnostic. You can have so one of the things that I didn't share is the way in which we built the schema. And by virtue of leveraging something really cool that Google made a long time ago called protocol buffers, we actually made it so that all of the fields in the schema are optional, which may sound like madness. However, the fact that you represent all of the knowledge utilizing and leveraging ontologies then allows you to standardize it. So there's nothing that says that by representing the organism that you want to represent and making sure that there's information about the taxon, you can build a phenopacket. I know that it's being used, and I should have shared this. I didn't put a slide, but it's being already deployed in the wild. And so, for instance, the Japan biobank AMED has already adopted, and all of their information is not represented. Phenotypic information is not represented in the shape of phenopackets. That's over 800,000 records, and it's I have not started to do it myself, and I have not heard of anyone that is trying to generate phenopackets with plants, but there's nothing that says that you couldn't. And as a matter of fact, we're about to publish a paper with our new Phenopackets tools, which are tools that allow for the programmatic generation of the packets and the validation as well. So you would be able to set the restrictions on how you would like to use them. And it's advantageous for being able to conduct analysis with those, building phenotypic profiles, building cohorts or groups of your biosamples. So I see potential in lots of good possibilities to use it.
So the biobank you talked about, is that all kind of animal or human
yes, that's
sample
all human
right now?
Yeah.
Okay.
But the biosamples database at EBI actually allows you to generate phenopackets as well. So, and I don't know if biosamples database has I think it has non-human data.
Because I think kind of the interchange of phenotypic data among the member databases of AgBioData that have some plant component would really be interested and could benefit from all the work that you have put in already. Thank you.
Yeah, I think that would be brilliant. Yeah. Sorry, go ahead.
All right.
That's it for me. Thank you.
Cheers. Good to see you.
There are some questions still coming up on
Yes.
the chat, so I'll try to start from the beginning here. So Sook is asking, who curates phenotypes into Phenopackets? Is it Monarch, or do you have other community members doing it? That kind of alludes to what Tanya was going in terms of, multiple inputs and how to handle a schema that's sort of as nicely broadly defined as you say, but there might be problems in the implementation aspect, right? So
we do have guardrails, right, to make sure that the freedoms are allowed. However, it doesn't go bunkers. And so that's why the leveraging the ontologies to represent the information is one of those guardrails. And so who curates the phenotypes? The researcher curates the phenotypes, and we're building validation tools so that you have a certainty that, against which you can run your data, so that you have certainty that you are correctly, accurately representing your phenotypic data into a Phenopacket. Our dream is of a world in which, for instance, the journal says, sure, why don't you submit your information and also represent all of the phenotypic data in the shape of Phenopackets, and then to have a centralized repository where people can actually compute over Phenopackets. That's why we are making them, right? Like interoperable, computable, case level or row level phenotypes that can be computed over. And so for us specifically, when we're working with Human Phenotype Ontology information for a specific case, then our team of curators does the work of turning curation data, annotation data into a Phenopacket. But it's on a case-by-case level, and there's not a centralized repository where you just take your data and somebody generates the curation for you. Does that make sense?
Sook, are you good?
Yes, thank you. Thank you.
Good to see you, Sook.
All right, there's more questions in the chat. Lenore said, I don't know if this is a question or comment, Lenore, maybe you can mention, curious about how we might be able to employ the ontology mapping to unify.
Yeah, SSSOM, you mean SSSOM? Yes. I mean, again, that's also species agnostic right? It's the concept of being the things that you have to take into consideration in order to conduct, build mappings across ontologies. So I would say, call Nico Matsensoglu, who's the primary author, but I think, we have the ability to triage a little bit and give a little bit of help desk sharing. There is something called the OBO Academy. Once every other week, I think it's once a month. Once a month, we come together as an interest group open to anybody, it's not Monarch specific, to learn all sorts of wonderful things about ontologies. And I'm pretty sure that and we record them and make them available. So I'm pretty sure there's a recording about using SSSOM. So Lenore, if you ping me, I will make sure that you get that recording. Yes, OBO Academy, Megan. This is the ontology. Oh my gosh, what OBO stands for? Biological ontology, sorry. It's for biological ontologies. And so, so we decided there's so many people interested in different aspects about ontologies themselves, how to build patterns, how to do the mappings, what does what does it mean to run a reasoner over my ontology, or what does it mean to axiomatize some concepts. And so we figured, we had enough questions that we felt empowered to start with Ontologies 101. And we've been at this for almost a year now. And there's a really good library of recordings that I can make available. So I can send it to Monica for distribution to the wider AgBio data. They're publicly available.
Great. Yes, please. Okay. I'll have to look into that myself too. Okay, Lenore had a couple more comments here. So are you doing anything to capture phenotypes from the literature? Is that done manually or computationally?
Yes, exactly. So both. So we would like it, instead of, this is the eternal fight of curating after the fact, or should there be a biocurator involved in the process of review, right? Like, this is me putting my ISB old hat. And so it happens at both levels. Right now, there isn't a process for that to be the case. But if we need it, then we will curate Phenopackets, the phenotype themselves, in order to be able to generate the associations before we put the publication out. So it's or depending, so for instance, we also do efforts like this. There's this incredible group of prenatal clinicians who were thinking, you know, the Human Phenotype Ontology is good and all, but your prenatal phenotypes are pretty gnarly. So why don't we sit down and review, because there are some gaps that we've identified. So for 6 months, we sat down and we would get into these like intense 2-hour workshops. Normally we would have done this in person, but of course COVID wouldn't allow it. So we got together and we did 6 of these 2-hour sessions with clinicians from across the globe. We had about 80 clinicians who would provide information and give us this breadth of knowledge about what's happening in each of the stages. And so we're talking, we had sonogram experts giving us information about all of the images. We had OBGYNs who are in the delivery process themselves. And then we were able to really create a much more robust branch of the ontology dedicated to the representation of abnormalities in utero. And we published this paper just last winter, or at the end of 2022. And it was a concerted effort of absolutely manual curation of phenotypes. And so what we do, because not every clinician is going to sit down and learn how to use Protege. I don't want to learn how to use Protege again. So what we do is we give workshops and we give them the tools, and then we tell them, never mind that this thing is called GitHub and is done for developers. This is what you click and this is what you tell me. And if you give me a name, a textual definition, and the generalized location of where this should exist, bam, then we can actually work with the ontology reasoners and then merge as necessary.
Great.
I have another question. So, like, how do you reach out? I guess you do those workshops, and how do you reach out to authors and clinicians so that they do put their findings into Phenopacket? Like,
Cheers.
also,
Yeah.
Also, like, do you work actively on reading publications to put the data into Phenopacket?
So as necessary, so the last part, as necessary for a study, then we would have curators go through papers in order to generate.
Oh, so you have specific project for a specific.
Right, exactly. So from our perspective, the need was there to be able to get phenotypic information at the road level in and out of the electronic health record, right? That was what did not exist. And there's lots of use cases for how the data was represented for clinical purposes, but not for research purposes. So we figured, here's what we do with the disruption. We actually generate a packet that can be used to compute over for research purposes. And so we focused on the generation of the schema itself and then the dissemination throughout different groups. For specific projects, then somebody would either get all of their data, massage it and turn it into the format, and then run a validator to make sure that the data comply to the schema, and then use it to compute over your millions of patients. So there's a project called the National COVID Cohort Collaborative that was born, of course, at the beginning of the COVID-19 pandemic and continues to work to this day. And they have used Phenopackets to compute over phenotypes from patients with long COVID. For the first time, about 80 or so hospitals in the United States are linked in an enclave that protects the identity of all the patients, of course, but you can generate Phenopackets from the phenotypes that are captured in those instances and then compute over, run machine learning, trying to find those links between the gene, the drug, the disease. And so for our case specifically, I don't sit down to curate them, but when people need it, they do. And the idea is that encourage people to curate them and deposit them in public repositories.
Mm-hmm. Oh, so where is the public repository then? Like
there isn't one, right? Like, so we don't have a centralized repository for Phenopackets right now. All of these projects, the European Joint Project on Rare Diseases, EGA, Japan Biomed, what's the one in the bird? I forget. They all have they've generated their own set of Phenopackets, and they have them in their own repositories for the purpose of running their own research. But we don't have one. We're talking, we are talking with NIH and are very enthusiastic about the reception that we have received from them in terms of actually implementing it in some of the institutes. And so maybe that becomes an avenue to explore. And then there was a first part to your question that I've now forgotten, Suk. Sorry.
I forgot to.
Sorry.
But great, thank you.
Of course, of course.
Great. Clay was just pointing out that there's a group working on this type of data in plants and had a link, so we should check that out.
Yeah.
Any more questions?
Oh, brilliant.
And yet, I might just mention, so in AgBio data, but we've had this RCN going on for a year now, and where we're trying to just figure out what the problems are and create solutions, not actually funded solutions, unfortunately. So they might be ideas and problems that are solidified that we can write a grant about. One of the things that came out from the Data Federation Working Group last year was that we are having problems with phenotypic data exchange, and that might be something that one of the things that we as a group would need to focus on for plants, possibly the largest component of taxonomic component of AgBio data first. So we should probably be looking into Phenopackets as a group. We're hoping to have a working group start out, but yeah, since it seems like we're filling a void, and that if this is what the biomedical databases are using, and if it fits the needs of our communities and is usable, then we will have to look into this, see if it's.
That's absolutely right, and not just because we know each other so well, but because by design it's a community effort. As you come across the information and start digesting, and you find, it would be really good if Phenopackets did X, then of course those are things that we can talk about. So right now there's another standard called a pedigree standard that is being, developed, and they said it would really be great if Phenopackets did this, and there's a branch now that is cultivating those requests, and so. And you know where to find me. So we can, have a sit down with a brainstorming session. We can create a workshop, or just, give you the information so that you can do it on your own and call us when you need that next step of, how do we work on implementing this at scale, or what strategies do we have for dissemination? Oh, and I think that was Suk's first question, like, how do you call the clinicians to come over? And so it's a very much a word of mouth. If I wanted to work on what is it that mosquitoes do when they get cold and sleep? What's it called?
Diapause.
Diapause. If I want to curate all the genes associated with diapause and phenotypes, I would call Monica. And so this is the same thing. You just like word of mouth, go and call in to the people that and then they spread the voice. We ended up having calls with these phenomenal clinicians on prenatal phenotypes from Israel who heard from somebody and showed up to the workshop, and it was great.
Fabulous. Yeah. All right. Any last questions, last words?
Well, my last words are a huge thank you, because this feels like quite the family reunion to see you all again, and to share a little bit about what I am passionate about right now. So thank you for the invitation.
Indeed. Thank you for coming. It's great to see you again and doing great and wonderful things in the biomedical community as well.
Cheers.
All right. Well, thank you all, and talk to you next time.